Hello guys, now we are continuing those short notes as we discussed in our previous video. Okay, now chloramine. This chloramine is used as chemical agent to disinfect in school labs. Okay, now autoclave sterilization is used for destroying isolated tetanus culture, culture. Okay, and how we can test that autoclave is working properly? Okay, so to test, basically use spore forming bacteria are used as test organism. Okay, now next point is about our gas gangrene is caused by clostridium perfringens, Clostridium perfringens. As we know about this Clostridium perfringens, they are anaerobic spore forming, and they are also gram positive. Okay, and what will be the first treatment? First treatment basically means before lab diagnosis. Okay, so what will be the first treatment before lab diagnosis is polyvalent specific serum. What is this polyvalent specific serum? This is basically, in this there are antibodies for many antigens. Okay, we have antibodies, AB means antibodies for many antigens. Okay, now our next point is blood examination is done for sepsis, how to check the person have sepsis or not. So we do blood examination. Okay, and repeating of infection after some time is known as reinfection. Okay, now in infants, this point is important. Okay, in infants, that BCG immunity is preserved till live bacterial strain is present. This means present. Okay, positive and anti. Present is present in body. Okay, and such immunity is non-sterile. Non-sterile basically means infectious. Okay, non-sterile means infectious. And see, as we know, this BCG vaccine. Okay, what is it? This BCG vaccine is live attenuated vaccine. What does it means? In infants means in children's. Okay, in children, this immunity is only present till that BCG vaccine, that strain, okay, that bacterial strain is living, is alive in that person's, in that infant's body, okay, in the infant body. Till then the immunity is present. Once that live bacterial strain will die in that person's body, such that immunity will be non-preserved, means not present or not continued, okay. That is only preserved till the strain is living, okay, that BCG vaccine strain that we put in the patient's body, okay. As we know, this is used for TB, tuberculosis, this BCG vaccine. Now the next is our Candida. This Candida is fungal disease, okay. This is usually gram positive. This is gram positive. They are oval, oblong cell, and they does what? Budding. BU. They does budding, okay, and they forms pseudo PSU DO pseudo mycelium, okay, forms pseudo mycelium, and plaque on, they're basically plaque like form, plaque on white exudates. White exudates are taken for diagnosis, okay, in case of oral candidiasis. Oral candidiasis. Plaque on white exudates are taken for diagnosis in case of oral candidiasis, okay. And what is the treatment? Treatment for Candida, that is nystatin, 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 okay. And we also discuss in our previous videos, okay. Now another point, subunit vaccine. What is this subunit vaccine? Okay, these are basically fragment of pathogen used, okay. And what type of fragment? Like basically it could be protein, peptide, or like any part, okay. Like we took any part of the pathogen and inject that into the person's body, and the immunity form against that fragment as an antigen, so your body will form immunity against that pathogen also, okay. The least allergic develops active specific immunity, okay. And this subunit vaccines, like we give in influenza virus, okay, like given in influenza, okay, like to prevent seasonal influenza, okay. And what we do, like for research work, so for research we do isolation and serological identification for that influenza, okay. Now differential diagnostic medium, okay. So basically it supports growth of any microbe, but still distinguishes them some or other way, okay. Like it is it support this differential diagnostic medium, it supports growth of any microbe but still distinguish them some or other way. Some or other way means like It will show some organisms have motility, give them different colors. Okay, so some or another way they will differentiate it from each other. Now our next point, if antibodies increases 4 times in paired serum, it means diagnosis confirmed. Okay, so if antibodies increases 4 times in paired serum, it means confirmation of diagnosed or diagnosed confirmed. Okay, now another one, dysbacteriosis treatment is eubiotic. Okay, now another one, like checking post vaccinal immunity. Okay, and this is also known as or retrospective study. Okay, is done by serological method. Okay, serological is basically that is antibody test. Okay, like after giving some vaccine, we have to check like that vaccine is working or not in the patient's body. Okay, in the person's body. So we does what? We took the blood out from the person's body and check their antibodies. Okay, and that is known as serological test. All right, so that's all in this video. Now we will continue these short notes in our next video. Thank you.